Hey guys, very warm welcome to this YouTube channel that is BioLog. Again, I'm Mosha Yadav and this lesson is about different type of plant tissue culture techniques. Okay, so on the basis of what kind of explant we are going to use in this plant tissue culture technique or what kind of tissue is extracting from the donor plant. So on that basis, we classify them into different type. Okay, so I already discussed in a previous video lecture that yes, plant tissue culture can be divided on the basis of their explant extracted from donor plant into different type. These are embryo culture, seed culture, meristem culture, protoplast culture, organ culture, pollen culture, callus culture, cell culture. Beside this, there is also too many type of plant tissue culture. So after this video lesson, we guys discussing one by one each and every type of culturing method that we are going to use in plant tissue culturing techniques. Okay, so this particular video lesson is about callus culture. So in this particular video lesson we are going to discuss about callus culture, that what is callus culture and how it is going to differentiate into the entire plant. Okay, so if we talk about the basic of this callus culture, then callus means unspecialized, unorganized and a dividing mass of cell. Okay, so if you discuss about this term, that what is callus, before dealing this callus culture you have some idea about callus. Whenever you talk about callus, then callus is a term that is we generally use for a bunch. Okay, generally for a group. Okay, for group of cells or group of tissue, or these group of cells and group of tissues are unspecialized and unorganized, that each and every cell of this callus can responsible for the formation of entire plant. Okay, so callus culture की single cells जो होती हैं they show totipotency. Okay, totipotency, you know what is totipotency? It is the ability of a cell that it can differentiate into the entire plant, into the entire organism. Okay, so whenever you discuss about callus culture, then there is a culturing technique in which we are using a callus that is extracted from the donor plant, and each and every cell of this callus is unspecialized, unorganized and a dividing mass of cell. Okay, so those cells have capability to behave like a totipotent cells, and also they have a capacity that they can divide, multiply and they can grow. Okay, because these are callus culture, bunch है हमारे पास, cell का bunch है. ठीक है, so callus is an irregular mass of parenchymatous tissue with meristematic loci. Okay, so if you consider your plant or a donor plant, then you can see that the meristematic portion is highly dividing portion of the plant. Okay, it has the capacity to divide, to multiply more and more as compare to your older part of your plant. Okay, so meristematic tissue जो होता है उसके अंदर highly amount में dividing capacity होती है. So whenever you talk about callus, then you can correlate it with the meristematic tissue. Just like your meristematic tissue of the plant, callus also have the capability to divide, to multiply. Okay, to grow. Okay, so callus is an irregular mass of parenchymatous tissue. Basically in callus culture we are using the bunch of cells, and these cells are specifically parenchymatous cell. Okay, you all read I hope you all of you know about parenchyma, collenchyma, sclerenchyma. Okay, so whenever we are dealing with the callus culture, then just keep in your mind this. Yes, callus culture consists bunch of parenchymatous cell. Okay, so example के लिए there is a tumor tissue that grow out of the wound of differentiated tissue and organ. Yes, whenever you see that अगर आपको कोई आपको इंजरी हुई है, ठीक है, इन दैट केस आपके पास क्या होता है कि डिफ्रेंशिएटेड टिश्यू एंड डिफ्रेंशिएटेड ऑर्गन में से भी वहाँ पे ट्यूमर टिश्यू जनरेट हो जाते हैं, एंड दीज ट्यूमर टिश्यू रिप्रेजेंट अ कैलस. ओके, अ बंच ऑफ सेल्स, अ अनऑर्गेनाइज्ड अनस्पेशलाइज्ड बंच ऑफ cells, okay, which have much more capacity to divide, to multiply. Okay, so for this type of tissue culture, the culture is often sustained on a gel medium. When you are performing, when you are dealing with callus culture, then this kind of culture media is generally sustained on a gel medium. Okay, so the nutrition medium you are carrying, or the culture media you are carrying during this callus culture, it must be a gel medium. Okay, so which is composed of agar and a mixture of given macro and micronutrients that depend on the type of cell. Okay, we already discussed कि yes, whenever we are dealing callus culture or any other kind of plant tissue culture, then we have to keep in mind that yes, the kind of the composition of nutrition media or different component factor depend that which kind of tissue we are extracting from donor plant, and which part of tissue is going to use as a culture. Okay, so on the basis of, on the type of cell, we are deciding that yes, we have to use this kind of nutrition media, we have to use these kind of growth factors. Okay, so on the basis of cell type, all of the nutrition composition is decided. Okay, so different type of basal salt mixtures such as moraceous and skog medium are also used in addition to vitamins to enhance growth. If you consider your callus culture, then you there is two kind of culture media which are suitable for this kind of culturing method for callus culture. These are murashig and skug medium. Beside this, you also add some vitamins, some essential vitamins and growth promoters like auxins and cytokines in the culture media that enhance your callus culture division capacity. Okay, all right, so you can see that here is a, you can see that easily difference in between callus culture and normal cell. Okay, so as I already told to you, okay, yes, callus culture or callus culture is just like your tumor cells. Okay, means a bunch होगा आपका cell का bunch होगा, dividing cells होंगी यहाँ पे, highly unspecialized cells होती हैं, undifferentiated cells होती हैं, right? So you can see that this image represent example of friable callus of wheat. This A image represent callus, यहाँ पे callus culture हो रहा है किसका, of wheat का, and this B image representing compact callus of mulberry. You can see that these are bunch of cells which are showing their irregular growth, undirectional growth, unorganized growth. Okay, you can see that yes, both of these kind of callus culture, this one is for mulberry and this one is for wheat, and these two images represent a callus culture. Okay, moving to the next slide, which represent the application of callus culture, that how callus culture is going to be useful for us. So it serves as a starting material to initiate single cell and suspension culture. Yes, whenever you are dealing with single cell culturing method of plant tissue culture or suspension culture, then you can see that the initiation point of suspension culture as well as single cell culture is formation of callus culture. Okay, so it can be we can consider it as a initiation point. So if we are going to deal with these two kind, then we have to follow callus culture formation. Okay, second application of this callus culture is that it provides system for various morphogenetic and physiological studies. Okay, so if you want to study some of morphological character, some of physiological character, then you can go through with the callus culture, because callus culture are unorganized cells, and with this callus culture you can easily separate a single cell and you can easily observe the morphogenetic changes from undifferentiated cell to differentiated cells. Okay, so you can trace out the morphological and physiological morphological and physiological changes that occur in single cell that is extracted from that callus culture when you are tracing out your morphological and physiological characters. Okay, so it also useful for producing secondary metabolites. A number of secondary metabolites are, essential for our daily uses, daily life. In that case, generally we perform callus culture to produce a high amount of secondary metabolites. So in this manner, callus culture is a best suitable method for producing secondary metabolite in high amount, in excess. Okay, it is also useful for generating useful somaclonal variations. You know that what is somaclonal variations? Okay, somaclonal means it, you can correlate it with your somatic cells variations, or different kind of changes that are occurring in somatic cells, and these changes are heritable changes. So these are genetic as well as epigenetic changes, or overall we can say that the total of changes or total of variations that can be formed from genetic or heritable level, genetic level पर भी आपके perform हो सकते हैं, and they can also be considered as epigenetic level. So whenever you are considering somatic cells changes, somatic variations in somatic cells due to genetic as well as epigenetic changes, then we can consider that these kind of variations are somaclonal variations, means variations in somatic cells are same kind of somatic cells due to epigenetic as well as genetic changes or variations. Okay, so these are overall applications of callus culture. So with this lessons, I hope you are able to understand the complete process of callus culture. What is the basic of callus culture, what are its important applications, and in which manner you can use this callus culture, because every and single topic has their advantages as well as their disadvantages. So you have to keep your advantages and disadvantages in your mind, so that you can easily understand the basic concept of that topic. I hope you guys like this video, and if you like it, then please share it, subscribe it. Thank you guys.